Good morning, children. I am Shubhadeep Ghosh and I will be taking your biology classes in this session. I hope you are all doing well amidst the severity of the current pandemic situation. I am thankful to the school authorities who have endeavored to conduct such online tutorial and classes. There are many students who have not got the opportunity to avail books, but it is my advice to you all to focus on the video sessions very carefully. It will be very much useful to you as you can go through them a number of times and find the recorded mechanism very much satisfactory. You will get interested in this and continue the classes regularly. Good morning, students. Today we will discuss about the cell division and the cell cycle. The ability of a cell to divide and reproduce itself is a major characteristic of most living cells. The process by which a mature parent cell duplicate into two daughter cells is called cell division. Cell division is of great importance in the life of an organism. Multicellular organisms like us start our life from a single cell structure called zygote. This zygote, after division, transformation, that is growth and development, and differentiation, gets converted into a fully formed organism. Cell division increases the number of cells and increases the surface area of the body. Cell division helps in wear and tear of tissues. It helps to repair them and replace dead cells. Cell division is essential for the continuity of life in the form of new generations. Cell division, as we know, is differentiated into three major phases: amitosis, mitosis, and meiosis. Amitosis is a direct cell division showing nuclear cleavage without the formation of spindle fibers. The process of cell division in which the nucleus first constricts in the middle to divide into two daughter nuclei, and then the cell undergoes changes without spindle formation and ultimately divides into two daughter cells. Amitosis occurs in microorganisms such as bacteria. Mitosis is the normal process of cell division of the somatic or body cells in most eukaryotic multicellular organisms and divides the mother cell into two daughter cells identical to the original. Mitosis occurs in somatic cells or general body cells of multicellular eukaryotes, except the nerve cell, the muscle cell, and the sex cells or gametes, that is, the sperms in male and ovum in females. Mitosis also occurs in the plants, in the growing tips like roots, stems, leaves, flower, apical and embryonic buds, and in cambium of plants. Today, we shall entirely discuss about the cell cycle and the interphase. In continuously dividing cells, an individual cell passes cyclically through two main phases of cell division, namely the interphase and the mitotic or M phase or cell division phase. The cell cycle is the entire sequence of events happening from one end of a nuclear division to the beginning of the next. You can see it is the entire cyclical events which is occurring continuously in a reproducing cell. Interphase is a stage between two mitotic cycles in an eukaryotic cell during which various physical and chemical changes for the preparation of cell division takes place. So, it is also sometimes referred to as the resting phase, but actually it is a phase of great importance where various changes, very important changes takes place within the cells. The activities going on inside a cell during the interphase state can be summarized as replication of DNA, synthesis of nuclear proteins, that is histones, division of centrioles, synthesis of energy rich compounds to provide energy requirement of mitosis phases, that is ATP, synthesis of proteins for formation of spindle fibers, etc. Major characteristics which occur during the interphase are the growth of both nucleus and cytoplasm occur to maintain the normal size of the cell. The nuclear membrane remains intact and distinct. Exact replication of DNA and duplication of the chromosomes from one chromatid, that is monad, to two identical chromatids, dyad, occurs. Enlargement of nucleolus due to the accumulation of RNA and ribosomal proteins occurs. In animal cells, a new centrosome is formed from the existing one with a pair of centrioles. The interphase is differentiated into G1, S and G2. G1, though the name may signify for growth, does not actually stand for growth. It stands for gap. G1 phase starts for post mitotic gap phase. G2 phase states pre mitotic gap phase, and S phase stands for synthetic phase. In G1 or post mitotic gap phase, the cell size increases. The RNA and various regulatory proteins are synthesized. Division of mitochondria and chloroplast occurs to provide energy. In G1 phase, the number of chromosomes, that is 2n, is equal to 46. Next, the successive stage, that is the S or synthetic phase, occurs, where the DNA replication takes place. The volume of nucleus becomes double. Duplication of chromosome from monad to two identical sister chromatids, that is dyad, occurs. Here the number of chromosomes becomes 92. The successive phase which occurs next is the G2 or pre-mitotic gap phase. Here the synthesis of specific RNA and proteins for spindle formation and cell division takes place. Organelles are duplicated, new organelles are formed as the mother cell, is going to divide into two identical daughter cells. This is the summary of the interphase phase, as the interphase is constituting only the G1, S and G2 phases. Now various changes takes place during the cell division phase. There must be something to check whether all the things are going accordingly or not. Hence, there are some checkpoints which are provided to the cell. The first checkpoint is the G1 checkpoint here at the end of the G1 phase. It checks if the cell size is proper or not, the proteins accumulated whether sufficient or not, and ATP is synthesized for the energy requirement or not. Here you can see another phase, that is the G0 phase. It is the arrested gap phase where the maturation and differentiation of cells which do not undergo the divisionary phase takes place. Another checkpoint lies at the end of the G2 phase, just before the beginning of the mitotic or cell division phase. The G2 checkpoint checks whether the DNA replication has been proper and complete, the organelles are duplicated or not, the protein accumulation and ATP synthesized for the requirement of the mitotic or cell division phase has been done or not. The last checkpoint, that is the mitotic metaphase checkpoint, lies at the metaphase stage, which we will discuss later on in the next classes. Thank you, boys, for today. Remain safe, stay at home, be healthy, and revise this work. Okay, here is a snapshot of the total board.